Mendel's predecessors had focused on entire plants. Mendel wisely chose to isolate a single feature at a time and follow it down the generations. He also resolved to count his results, something that had never been done before, and to look for patterns in the numbers he came up with. Mendel searched for the ideal experimental model and found it in the common garden pea plant. It has a number of traits that come in two contrasting forms, which make them easy to spot and categorize. For instance, the pea pods are either full and smooth or shrunken and wrinkled. Stems are either short or tall. Its flower has a shape that protects against contamination by stray pollen. And lastly, it self-fertilizes. Its pollen fertilizes eggs within the same flower. This allows it to produce seeds which breed true, meaning each offspring plant is exactly like its single bisexual parent. Over the following 8 years, Mendel planted an estimated 28,000 carefully controlled crosses between different varieties of pea plants. He explored every possible combination, sometimes for up to 7 generations, and meticulously counted the results. One of the contrasting features that Mendel studied was the color of the plant's flower. One of his true breeding varieties always grew purple flowers, another always grew white ones. Their seeds formed the parent or pea generation. As soon as buds appeared on the vines, Mendel carefully opened them and removed the stamens to prevent self-fertilization. He later took pollen from purple flowers and sprinkled it on the pistils in white flowers. He also did the reverse. To prevent contamination by wild pollen, he then wrapped the flowers with protective bags. As a further control, he moved some of the plants into the insulated environment of a greenhouse. Weeks later, both groups of cross-fertilized plants developed seeds. Mendel stored the hybrid seeds over the winter and planted them the next spring. When the plants in this first hybrid or F1 generation grew flowers, they were all purple. Mendel called this trait the dominant one. He termed the trait that disappeared in the F1 generation recessive. Mendel let these F1 hybrid plants self-fertilize and produce new seeds. The next year, Mendel planted the F1 seeds, producing the next or F2 generation of plants. This time, there were some white flowers mixed in among the purples. He was well aware that traits sometimes skipped generations, so this was no surprise. But Mendel discovered something that no one else had ever noticed before. He counted the number of white and purple flowers. In similar fashion, he counted the results of crossing 6 other contrasting traits through to the F2 generation. When he worked out the ratio of dominant to recessive, in each case it came very close to 3:1. Mendel had uncovered a pattern of inheritance, something that had eluded everyone from Aristotle to Darwin. This was a great discovery in itself, but Mendel also wanted to explain it. Using just his data and his imagination, Mendel came up with a model of how heredity works that remains the cornerstone of genetics. He concluded that life is not transmitted in one piece, as most people assumed, but in discrete particles, which he called factors, and which we now call genes. Using his particle model, Mendel was able to explain the great paradox of heredity, how both inheritance and variation can coexist, how like can beget both like and unlike. Mendel concluded that these invisible factors are passed along unchanged, which accounts for inheritance. but that sexual reproduction leads to new and different combinations of them, which accounts for variation. Mendel theorized that a trait is determined by not one, but two factors or genes. These factors also come in dominant or recessive forms. But Mendel made an important distinction between the nature of the trait, which we call its phenotype, and the nature of the underlying pair of genes, which we call its genotype. For his experiment with purple and white flowered plants, there were two possible phenotypes, dominant purple or recessive white, and three possible genotypes, two dominant genes, in which case the phenotype is purple, two recessive genes, in which case the phenotype is white, and one of each, in which case the single dominant gene causes the phenotype to be purple.